Hello, this part will be short because I will just read on neutrophils and cell signaling because I will have a separate video for forms of shock so that it will be easier for you to remember and to navigate the types of shock. Okay, so let's proceed to the neutrophils. Neutrophil. Neutrophil activation is an early event and in the upregulation of inflammatory response. Neutrophils are the first cells to be recruited to the site of injury. Polymorphonuclear leukocytes, or PMN, remove infectious agents, foreign substances that have penetrated host barrier defenses, and non-viable tissue through phagocytosis. However, activated PMNs and their products may also produce cell injury and organ dysfunction. Activated PMN generate and release a number of substances that may induce cell or tissue injury, such as the reactive oxygen species, lipid peroxidation products, proteolytic enzymes such as elastase and cathepsin G, and vasoactive mediators, leukotrienes, eicosanoids, and platelet activating factor. Oxygen-free radicals such as superoxide anion, hydrogen peroxide, and hydroxyl radical are released and induce lipid peroxidation, inactivate enzymes, and consume antioxidants such as glutathione and tocopherol. Ischemia reperfusion activates PMN and causes PMN-induced organ injury. In animal models of hemorrhagic shock and mortality, a neutrophil depletion prevents pathophysiologic sequelae of hemorrhagic and septic shock. Human data corroborate the activation of neutrophils in trauma and shock and suggest a role in the development of MODS. Plasma markers of PMN activation, such as elastase, correlate with the severity of injury in humans. Interaction between endothelial cells and leukocytes are important in the inflammatory process. The vascular endothelium contributes to the regulation of blood flow, leukocyte adherence, and the coagulation cascade. Extracellular ligands, such as intracellular adhesion molecules, vascular cell adhesion molecules, and the selectins, such as E-selectin and P-selectin, are expressed on the surface of endothelial cells and are responsible for leukocyte adhesion to the endothelium. These interactions allow activated neutrophils to migrate into the tissues to combat infection, but also can lead to PMN-mediated cytotoxicity and microvascular tissue injury. So that's it for neutrophils. Next is cell signaling. A host cellular changes occur following shock. Although many of the intracellular and intercellular pathways that are important in shock are being elucidated, undoubtedly there are many more that have yet to be identified. Many of the mediators produced during shock interact with the cell surface receptors on target cells to alter target cell metabolism. These signaling pathways may be altered by changes in cellular oxygenation, redox state, high energy phosphate concentration, gene expression, or intracellular electrolyte concentration induced by shock. Cells communicate with their external environment through the use of cell surface membrane receptors, which, when bound by a ligand, transmit their information to the interior of the cell through a variety of signaling cascades. These signaling pathways can subsequently alter the activity of specific enzymes and the expression or breakdown of important proteins, or affect the intracellular energy metabolism. Intracellular calcium homeostasis and regulation represents one such pathway. Intracellular calcium concentrations regulate many aspects of cellular metabolism. Many important enzyme systems require calcium for full activity. Profound changes in intracellular calcium levels and calcium transport are seen in models of shock. Alteration in calcium regulation may lead to direct cell injury, changes in transcription factor activation, and alteration in the expression of gene important in homeostasis. and the modulation of the activation of cells by other shock-induced hormones or mediators. So, ganun pala ka-importante si calcium. Okay. To continue, a proximal portion of the intracellular signaling cascade consists of a series of kinases that transmit and amplify the signal through the phosphorylation of target proteins. The oxygen radicals produced during shock and the intracellular redox state are known to influence the activity of components of this cascade, such as protein tyrosine kinases, mitogen-activated kinases, and protein kinase C. Either through changes in these signaling pathways, changes in the activation of enzyme systems through calcium-mediated events, or direct conformational changes to oxygen-sensitive proteins, oxygen radicals also regulate the activity of a number of transcription factors that are important in the gene expression, such as nuclear factor Kb, apetala-1 or APETALA1, and hypoxia-inducible factor 1. It is therefore becoming increasingly clear that oxidant-mediated direct cell injury is merely one consequence of the production of oxygen radicals during shock. Okay. The study of the effects of shock on the regulation of gene expression as an important biologic effect was stimulated by the work of Bachmann and colleagues. The effects of shock on the expression and regulation of the numerous genes and gene products has been studied in both experimental and animal models and human patients. These studies include investigation into single genes of interest as well as large-scale genomic and proteomic analysis. Changes in the expression are critical for adaptive and survival signaling. Polymorphism in gene promoters that lead to a differential level of expression of gene products are also likely to contribute significantly to varied responses to similar insults. In a recent study, the genetic responses to traumatic injury in humans or endotoxin delivery to healthy human volunteers demonstrated that severe stresses produce a global reprioritization affecting greater than 80% of the cellular functions and pathways. The similarities in genomic responses among different injuries revealed a fundamental human response to stressors involving dysregulated immune responses. Furthermore, in traumatic injury patients, complications like nosocomial infections and organ failure were not associated with any genomic evidence of second hit and differed only in the magnitude and duration of this genomic reprioritization. Again, genomic reprioritization. So that's it. We are now going to proceed in the next video for the forms of shock, and we will first read the hypovolemic or hemorrhagic shock. Okay, see you there. Bye.